Hi, my name is Natalia Maryniak and I'm a PhD student here at DTU, National Food Institute, in the research group for food allergy. Now I would like to take a few minutes to show you this piece of equipment that we have as part of our research under FOODHAY. FOODHAY is the Food and Health Open Laboratory, which is a national Danish initiative and is founded by the Ministry of Education and Science. This piece of equipment is an analytical liquid chromatography system called ActaPure from Zofia. It is a fully automated equipment for small and large sample volumes from microliters to liters and can be used both for preparative chromatography in order to isolate and purify proteins from large volumes and for analytical chromatography in order to analyze the components of the mixture and their proportions. This equipment allows to isolate and separate proteins based on their physicochemical properties, such as polarity, size, or specific binding affinities. This means that you could, for example, use this equipment to isolate and purify proteins from plant or animal sources, monoclonal antibodies, and bioactive peptides. It consists of the solid phase, which is the resin in the column, and the liquid phase, which is the buffer. The sample is injected using a syringe in here, and the pump system delivers the sample and the buffer in the column. Different resins are used for different mechanisms of separation and also depending on the protein properties. In general, proteins of interest will bind to the resin in the column while others will flow through. Changing the buffer composition will break the interaction between the proteins and the resin, and fractions run through the column can then be collected in the fraction collector for further analysis. The separation is monitored using a UV detector located in here. In Food Allergy Research Group, we use this equipment in order to purify proteins in a small scale from plant or animal sources in order to determine the protein's profile and their modification upon processing. This is relevant in our research for alternative protein sources and correlating their physicochemical properties with allergy or tolerance-inducing capacity. Therefore, it contributes to the research in sustainability and food safety. Now I would like to show you an example of the results from my study. This is a chromatogram of my product of interest. It consists of different peaks representing proteins or protein aggregates. So first, I loaded my product of interest in a specific concentration and volume into the size exclusion chromatography column in order to separate proteins in the product based on their size. The chromatogram is shown as an elution time against the absorbance in 250 and 280 nanometers, representing an absorbance of peptide bonds and aromatic amino acids respectively. The principle of size exclusion chromatography is that the bigger protein are eluting first, so here you can see the biggest protein in my product, and over the time smaller proteins are eluting. Thank you for spending a few minutes with me looking at the chromatography system called ActaPure. If you are interested in using this piece of equipment, or you are in doubt whether this piece of equipment is suitable for your research purpose, then please contact Anna Sancho, a senior researcher in our food allergy group responsible for protein analysis. Contact information is shown on the screen.